Welcome to this episode of the Biology of Trauma podcast. I'm your host, Dr. Aimie. We'll be answering the question, how can we change our biochemistry to change our trauma responses? In this episode, we are looking at thoughts and behavior through hormones, metabolism, and biochemistry. I have brought in a distinguished guest for this conversation, Dr. Robert Lustig, a pioneering neuroendocrinologist. Now, a neuroendocrinologist is someone who studies and works with the intersection of the nervous system with the endocrine system, neuroendocrinology. And the endocrine system is all about hormones. So his work has been instrumental in understanding metabolic disorders and their role in our stress and mood. He had a big influence on me as I came into functional medicine in search of answers for my own health issues during my surgery residency. And when I looked at his work, it was fascinating to me, given that I had a master's in biochemistry, and I could see how our behaviors and thoughts all come from our biochemistry. It helped me make sense of what I was experiencing in my own physiology, health, and mood at that time. We finally met in a polyvagal study group where we both attend with Dr. Steve Porges, and we are both speaking at the Integrative Medicine for Mental Health Conference. So I took the opportunity to invite him to share something important with you, something that you're not hearing from other neuroscientists right now. My favorite quote from this episode is near the end, where he says, "The single nugget of truth that we must deal with here is that all behavior is really biochemistry." In this episode, you will learn whether the global epidemic of chronic health problems are a result of separate issues or part of a single, larger root issue in our stress resilience and physiology, the 4 breaks of the amygdala in fear conditioning, how our metabolism influences serotonin and our stress and mood regulation, the intersection of metabolism, cortisol, and insulin on depression, and finally, how our thoughts and actions are created at the cellular level by biochemistry and proteins. I am so very excited for this episode. Let's get started. First of all, welcome. And then you've been doing some great, fascinating work on really looking at the manifestations of our global chronic health problems and whether they are a result of separate solutions that each have a separate problems with a separate solution, or whether they are more part of a general maladaptation of our stress resilience and our physiology. So in the paper that you're working on now, for example, you talk about the syndemic and this idea that there is a syndemic health crisis. What do you mean by that?
Aimie, thank you very much for having me, and thank you all for taking the time to listen to it. So I don't know if you've heard of this term syndemic. I have to tell you, I had not heard of it until 2018 when a paper was published in The Lancet that, talked about the syndemic. And the syndemic is our systemic health crisis, our mental health crisis, and our general societal health crisis. So from the systemic health crisis, we have, of course, type 2 diabetes, hypertension, dyslipidemia, cardiovascular disease, cancer, dementia, fatty liver disease, polycystic ovarian disease. I might add all of those are chronic metabolic diseases, and they are all driven by defects in the mitochondria. We also have this mental health crisis of addiction, depression, post-traumatic stress disorder, suicide. The list goes on. I don't have to, belabor that. You understand that. The question is, what's the root cause of those? And it turns out those are all metabolic diseases as well. And they are all due to defective mitochondria in the brain. And then we have these societal health crises of xenophobia, war, climate change. Well, actually, those are the same. They're just not individual mitochondrial disorders. They're systemic, global, societal mitochondrial disorders when you actually get down to the nugget of them. So the question, that I asked several years ago was, are these isolated issues or are these somehow related? And if they're related, does one cause the other, or are they all epiphenomena from some other antecedent? And the amygdala, as I'm sure all of you listening know, is really important and, is at the heart, is really ground zero for all of these problems that we have. That stress, plays an outsized role in both the systemic and the mental and the societal health crises that we all face. And the question, of course, is, what happened to the amygdala? And it turned out nothing happened to the amygdala. The amygdala is actually doing what it's supposed to be doing. What has happened is that there are brakes on the amygdala. There are four brakes on the amygdala. And our review of the literature, our neuroscience, all the, taking all the imaging data, all the biochemical data, all the epidemiologic and demographic data, all the interventional data that's out there in the literature. And we sifted through all of this to construct this model. And what we realized was the amygdala is actually not broken. The amygdala is doing its job. What's broken are the four brakes. So think of it this way. You got a car. Okay, and the car has four wheels and each wheel has a brake, so you got four brakes. Well, there are four brakes on the amygdala, and all four are failing right now. Now, they're failing for different reasons, but they're all failing. And because of that, we have basically lost our ability to basically be introspective. We have lost our ability to be pro-social. We have lost our ability to be pro-collaborative. We have basically, gone into alone mode. And you see it in our systemic health issues. You see it in our mental health issues. You see it in our societal health issues. So what are the four brakes? First one, prefrontal cortex reasoning. And what's damaging that? Both dopamine and cortisol. Next one, hippocampus, memory. And what's damaging that? Primarily cortisol, but also glutamate. Next one, interoception. That is the information you get from inside the body coming up to the brain. And what's damaging that? Well, changes in serotonin, changes in anterograde neurotransmission, changes in inflammation, due to changes in the intestine, which are systemic. And finally, number 4, oxytocin, which is safety. Okay. And it turns out that the oxytocin receptor is the most methylated receptor in the brain and in the body. And when it's methylated, it's broken. It will not transduce an oxytocin safety signal. And so all you experience is threat. And in the face of threat, basically, the amygdala can't do its job. So it turns out there are serotonin links, there are dopamine links. We have gone from a fear reinforcement paradigm, which carried us forward through eons of evolution, into now a chronic stress paradigm, where basically any external stimulus is interpreted as threat. And the question is, what happened to, allow for that change? And of course, that's the loss of, stress resilience. That's what you all talk about. That is trauma. So what we're doing is we're basically piecing out the neuroscience of the pathogenesis of trauma. And it's 4 parts. That's why there's not one fix. There's 4 fixes. And we're, by doing this work, by developing this model, we can actually apply different interventions in silico in the computer to determine what will work and what won't work before it's actually even tried, and to determine whether or not you need combinations thereof in order to be able to solve individual and ultimately societal problems. So that's kind of what we did. And, we're very proud of it and writing it up now.
And as we look at the role of the amygdala, it sounds like what you're saying is that there's this state of the nervous system, and our neuroception has conditioned our amygdala to see fear everywhere. Yes. Rather than maybe perhaps the step where amygdala is creating the fear all by itself. It's like, no, we've actually got this neuroception, and it's the perception of constant fear that's programmed and conditioned our amygdala to have these constant fear responses and perceive everything as dangerous.
Well, I mean, that's one way to put it. It's a little bit more complicated than that ultimately, as you can imagine. It turns out the amygdala, as I said, is actually not the problem. The amygdala is doing its job. There's another organ in the brain outside the amygdala, but it's functionally connected to the amygdala. And it turns out it's the gate. And so it may not be the amygdala's fault. It may be the other parts of the brain's fault. But ultimately, the goal of the amygdala is to try to figure out how to score. Well, the more damage is being done elsewhere, the more the amygdala can't do its job. And that's what we're seeing. And so there are different phenomena that are going on. Now, when the amygdala processes any external stimulus, it sends the information to the bed nucleus of the stria terminalis. And from there, the bed nucleus of stria terminalis has to decide, is this an acute, manageable stress that ultimately requires learning, in which case it sends the information to the ventral tegmental area, which then gives you a dopamine response. And that dopamine then feeds back on the amygdala to learn. Like, for instance, when you put your hand down on a hot stove when you're 3 years old and you realize you're never doing that again. Okay, that's standard fear conditioning. That's what got us to, this level of evolution, and, societal capability, is that level of fear reinforcement. Or is it overwhelming, and is it going instead go to the locus ceruleus, which is then going to activate the sympathetic nervous system and, of course, the hypothalamic pituitary axis and generate that cortisol response? And that would be chronic toxic stress. Well, it turns out that bed nucleus of stria terminalis has an important job. Gating. It's a gate that ultimately determines which way the information is going. Well, you can overwhelm that BNST with chronic information, input, or with overwhelming input. Both of those, basically, it's like, the bucket that you overwhelm. And now the BNST can't manage it. And so the information ends up being shuttled to the locus ceruleus and the HPA axis. So which way the information goes is very much dependent on the type of information, the volume of information, the amplitude of the information, and the chronicity of the information. And the other thing that we learned from the work, from putting this model together, is that amygdala is malleable. It's not fixed. It undergoes something called synaptic weighting. And, this is well known from experiential, studies in the amygdala, that basically the amygdala changes. And it changes under the influence of the neurotransmitters that are inside it. And what are those neurotransmitters? Well, two of them are the serotonin 1A receptor and the 2C receptor. And they're basically changing how the amygdala interprets that information. And that's coming from your diet. So your diet plays a major role in how your amygdala transduces external stimuli. So this is how your interoception, is how your afferent vagus ultimately impacts on this as well. And you see this in terms of transduction of the pain signal. You see this in terms of transduction of the agony signal, both whether it's physical agony or mental agony. Okay, that it becomes much worse in the face of serotonin deficiency, which is, part and parcel of the systemic health crisis. Metabolic syndrome is in part serotonin deficiency. So you can see that these different, things that are happening to us all have the amygdala at the center. And by, understanding the inputs and the outputs, we can actually understand what we can do to try to mitigate and attenuate these, abnormal responses that we currently have. That's the idea of the project. That's the idea of the model. And, that's what I'm presenting to you now.
It's exciting. And when I hear you say the word overwhelm, for me, through that biology of trauma lens, that's a key word. There's that stress that can be a helpful stress, and we can adapt to that. Like we know the amygdala is always adapting. And then there's just overwhelm of information and overwhelm of inflammation at times. And yes, I don't think many people are thinking that their interoception, or their ability to connect with their body and notice those internal sensations, can be related to their diet at all. And yet you have found that neuroinflammation connected with the gut inflammation is playing a central role in this ongoing fear conditioning with the amygdala. So is that involving some of the glutamate with the hippocampus? How all do you see that gut inflammation affecting neuroinflammation and affecting the fear conditioning?
Well, so first of all, cortisol is a big, player in this, because cortisol causes hypermetabolism. Okay, that's basically what cortisol does. It causes all neurons to increase their need and utilization of ATP. Okay, and if the ATP is coming, great. You can then basically withstand the onslaught of the trauma or the stress. But if you don't, if there's something actually inhibiting mitochondrial ATP generation, like for instance, chemicals in the environment, chemicals in the food. My favorite, of course, is fructose, because fructose inhibits 3 enzymes involved in normal mitochondrial function. It inhibits AMP kinase, which is necessary for mitochondrial biogenesis. It inhibits ACAD-L, which is necessary for fatty acid oxidation. And it inhibits CPT1, which is necessary to regenerate carnitine in order to shuttle fatty acids into the mitochondria for oxidation in the first place. So if you have defective mitochondria, you have a defective amygdala. And if you have a defective amygdala, guess what? You have all the things we've just been talking about. So food matters a lot. And we have plenty of data to show that ultra-processed food consumption causes insulin resistance in the brain, which promotes depression, and which ultimately promotes metabolic syndrome, at the peripheral level, but also because of the cortisol, driving hypermetabolism. So this is a vicious cycle. This is a feed forward cycle. And, this first became apparent to me back in 2003. So 21 years ago now. Anybody know the name of Mary Dahlman? So she was a professor of physiology at UCSF. She was a crotchety old lady with a very sharp tongue, but she was friggin' brilliant is what she was. And she was the first person to understand this concept of the hysteresis loop. And she published a paper back in PNAS in 2003 on basically not the amygdala per se, but on the stress pathway and cortisol, and the fact that it was a feed forward cycle instead of a feedback cycle. And how comfort food mollified that feed forward cycle in order to basically foment metabolic syndrome. So Mary was a colleague of mine for many, many years. And 20 years ago, my colleague here at UCSF that I'm writing this next book with, Dr. Alyssa Epel, who perhaps you know and know of, she's the one who figured out that chronic caregiving shortens telomeres. So alters cellular aging, in part because of the cortisol, but, other reasons as well. So Alyssa and Mary and I locked ourselves in a room at UCSF. For 8 hours, we held a little mini retreat, and we didn't let ourselves out of the room until we had basically come to a codification of the different aspects of metabolic syndrome that we brought to the table. And I can sort of sum it up in a Venn diagram, actually in a Punnett square, okay, a 2x2 matrix, right? And this will be very important, I think, to your audience, this concept. And I still believe this concept is correct. So imagine you have a 2x2 matrix, and you put the word cortisol on the left side of the matrix, and you put the word insulin on the top of the matrix, okay? And you put a minus and a plus, and a minus and a plus. All right, so you have 4 boxes, 2x2 matrix. So you have cortisol and insulin as the 2 variables, and you have low and high. So the first box, low cortisol, low insulin. You can call that normal. Okay, next box, low cortisol, high insulin. We can call that simple garden variety obesity. Okay, high insulin drives fat storage. This is work that I've done for the last 25 years. Next box, high cortisol, low insulin. That is major depressive disorder, MDD. That's your melancholic depression. That's your weight loss depression. Okay? But you also know that even though you're losing weight because you're anhedonic, you're actually gaining visceral fat. You're losing subcutaneous tissue, but you're gaining visceral fat. If you stick somebody with melancholic depression in a scanner and you look at their insides, they're actually increasing their visceral fat because of the cortisol. And then finally, the last box, which is the most important box, high cortisol, high insulin, both at the same time. And that is the interesting box. It depends on who sees the patient first. If the endocrinologist sees the patient first, we call it metabolic syndrome. If the psychiatrist sees the patient first, we call it atypical depression. And the reason is because melancholic depression causes weight loss. Atypical depression causes weight gain. And the reason is because the patient's trying to mollify the depression. They're trying to mollify the hypermetabolism of the cortisol with food intake, with trying to increase their ATP by increasing energy substrate. And so that concept of this high cortisol, high insulin is what we're all experiencing now. And the high insulin is partly because of the food supply, and the high cortisol is for all the reasons we've discussed, the 4 breaks. But the bottom line is that's what we got, and that's what has to be fixed. So understanding how these different aspects of medicine sort of fit together was, part of the thing that drove this project of the amygdala. And how important the amygdala is, because it's the generator of the cortisol. There is no cortisol without the amygdala.
And I think that this is some of the work that is so needed, because it's different than what our models have been, but yet this is pivotal for then applying solutions and strategies. Because if part of the problem is that the cells are being driven by cortisol and need more ATP, but our mitochondria are compromised because of our diet, dysfunctional, not able to meet that demand, then that's a point that we can provide some solutions as well. I know that part of your work has been looking at the nutrition and the nutritional aspect of tryptophan and how that then feeds into the serotonin system, and how the amygdala dysfunctional oftentimes has that low serotonin. So I would love for you to touch base on where does that serotonin and tryptophan and nutrition piece come together in this view of the amygdala? Right.
So as I said, the amygdala has both 1A and 2C receptors, serotonin 1A and serotonin 2C receptors. And they're, coming, the serotonin is coming from the dorsal raphe. Okay. But it's also coming from the nucleus tractus solitarius. So coming from 2 places. Okay. And the dorsal raphe is the standard place where we, think of serotonin coming from, but the NTS also sends information, and it's calming down the amygdala. The NTS is extraordinarily important in keeping amygdala output down. And one of the things that does that is serotonin. Serotonin keeps that amygdala sort of under wraps. It's one, like I said, it's one of the brakes, the interoception. All right. So where's the serotonin coming from? Well, it's coming from two places. It's coming from serotonin being made de novo in the brain from tryptophan. Remember, tryptophan is the only source of serotonin. There is no other. Okay, you have to basically, tryptophan to tryptophan hydroxylase takes you to 5-hydroxytryptophan, and then it's, dopamine beta decarboxylase to get you to serotonin. So there are two steps, but basically decarboxylating that 5-HTP to become serotonin, 5-hydroxytryptamine. Well, when you ingest tryptophan, okay, it goes from the gut to the liver, and then the liver does something nasty. It can convert that tryptophan. Remember, tryptophan is the rarest amino acid. It is the amino acid that is the basically rate limiting amount of all the different amino acids. It's the one that we have the least amount in our diet. You get it from eggs, chicken, fish, not exactly ultra-processed food. Okay. That tryptophan goes to the liver. And if that liver is insulin resistant, then the liver converts that tryptophan to another compound called kynurenine, and it never becomes serotonin. It basically takes the serotonin off the table and makes you serotonin deficient. So you become tryptophan deficient because you're insulin resistant. What we've also learned from this work, by the way, one of the things that I'm sure you discuss in this group is the difference between depression and PTSD. To this day, no one understands the difference. What is the actual reason why some people get depressed, some kids get depressed, and some people get PTSD. And of course, there's overlap between the two. There are people, some people have both. And, so the question, of course, is, what's that about? And we think we know the answer to that. We think we've actually determined what's happening in the amygdala that's allowing for that. And the reason is because PTSD is low cortisol and depression is high cortisol.
Where would you start? Like, where would you say to someone who's sitting in front of you, given all of that, here is a simple thing that you can start doing today. Here is a simple solution that you can start with today, knowing that this is a big mountain that's going to take some time to unravel all of the pieces involved.
Right. And there are four things. There are four basic principles. Okay. You have to feed the gut, you have to protect the liver, and you have to support the brain. Okay. How do you do that? Number one, get rid of the sugar. Number two, add fiber. Number three, add omega-3s. Number four, get rid of the emulsifiers, because they're what's causing the GI and therefore the neuroinflammation. So those are the four things that have to be done. Get rid of the sugar, add fiber, add omega-3s, get rid of the emulsifiers. Now, those four things right now are ultra-processed food. That is the definition of ultra-processed food, those four things. And ultra-processed food is not going away. As much as I wanted to, it's not going away. But every country that has adopted an ultra-processed food diet now suffers from the syndemic, the systemic health crisis, the mental health crisis, and the societal health crisis, because the food matters. And the food has changed, and therefore the amygdala has changed.
And I feel like we can take care of the rest. We're applying the somatic work, the trauma work, and the parts work. And knowing this strong biological component is really a game changer, because without that, if we're still not feeding our gut, helping our liver, if we're still causing mitochondrial damage, I mean, what progress can we expect our neurons to make, our amygdala to make when it's still getting that information from inside, from our own biology?
Right.
And so,
perhaps that, Aimie, that, for your audience, this is perhaps the single nugget of truth that we must deal with here. Okay. And that is that behavior is really biochemistry. Every thought is a protein phosphorylation. So you can affect thoughts by affecting protein phosphorylation. Well, you can affect behavior, you can affect response to trauma by affecting biochemistry. And if you understand what the biochemistry is and where the biochemistry comes from. And, as a reductionist, as a scientist, as a physiologist, as, somebody who's spent a lot of years in the lab, to me, that is what makes sense. Not that there's anything wrong with talk therapy. Okay. You need that too. But until you actually change the biochemistry of the brain, I don't see us really making much headway. For instance, we've known about the neurotransmitter hypothesis since 1982, okay? We've known about Prozac since 1986, and the depression problem has only gotten worse. That ain't the way to fix it, okay? That's trying to solve the problem downstream of the problem, not upstream of the problem. We have to affect the root cause. What's the root cause? Well, the root cause is what I've just been discussing.
And that is your mic drop for the day. All behavior is a phosphorylated protein. All behavior is biochemistry.
All thoughts are phosphorylated.
All thoughts. Yes. And that I think is the perfect summary of the whole biology of trauma lens, where it's never going to be enough just to do something like talk therapy, because this is all biochemical. It's all happening at the physiological level. So thank you. I feel like this was a fire hose back into medical school, which is a wonderful, refreshing experience for me. I miss that. I miss that element of getting into this biochemical level, and really want to appreciate you for your work in the world, Dr. Lustig. Really want to appreciate you for that.
That's my pleasure, Aimie. Thank you.
Thank you for tuning into this episode of the Biology of Trauma podcast. The main takeaway from this episode is that all behavior is biochemistry. And this is why it will never be enough to just do talk therapy. Remember my key line: trauma becomes our biology, not just psychology. This episode is a great window into why we must incorporate the biology piece into everyone's healing journey. If you are curious to learn more about biochemistry, mood, stress, and trauma, then I have resources for you. First, you will want to have my guide to the 3 most common biochemical imbalances. These are ones that I find in those with persistent trauma responses and mood dysregulation. Until these are identified and supported with biology trauma repair tools, they will hold you back in depression, anxiety, even fatigue and pain. If you know my story, you will know that I discovered that I have all 3. So no wonder I was stuck in developing worse symptoms of trauma biology over time, like chronic fatigue, chronic pain, autoimmunity. I also have related podcast episodes if you are curious for more. And these would be episode 18, What Truth Do Trauma Professionals Need to Know About Grains with Dr. Peter Osborne. Episode 41, Solutions for Low Serotonin and GABA in Trauma with Trudy Scott. Episode number 2 with Dr. Peter Kahn on brain inflammation and its role on mood, anxiety, and the freeze response. I also have a few YouTube videos on this topic of biochemistry. I have one on the role of mitochondria in trauma work with Dr. Gabor Mate. I also have a video on the copper to zinc ratio you need for healing from trauma. If you could please take a minute to leave a review for this Biology of Trauma podcast. As you know, I am here to save you time and money by bringing you practical information that will help you accelerate the trauma healing journey. Your reviews and feedback are how I know how to improve this podcast for you, continue bringing you great content, and help others find information that could change their life as well. I'm your host, Dr. Aimie. Until next episode, lots of love.